Hey everybody, Ronnie here. So, I did a thing. I wrote a book, How Evolution Works. This is the third part in this series, and today we're going to jump into chapter 3, which is the role of water. This is basically the final setup chapter before we get into creepy crawlies. So, I hope you enjoy this. This is how evolution works. You can find this on Amazon in Kindle format as well as paperback. You can also now find it on Audible as an audiobook. Just look for How Evolution Works by Ronnie Joubert, and you will find this. If you like this content, make sure you give it a thumbs up. Don't forget to hit the subscribe button and enjoy How Evolution Works Part 3, the role of water.
In the ancient and primordial landscape of early Earth, a single water molecule embarked on a journey that would illustrate its indispensable role in the unfolding story of life. This molecule, composed of two hydrogen atoms covalently bonded to an oxygen atom, was part of the vast and turbulent ocean that covered much of the planet's surface. As it drifted through the water, it participated in the intricate dance of chemical reactions that laid the groundwork for the emergence of life. The journey of our water molecule began in the vast expanse of the early ocean, a body of water teeming with simple chemical compounds. The ocean's surface was constantly agitated by the energy from sunlight, wind, and geological activity. This incessant movement facilitated the mixing of gases from the atmosphere with the minerals and organic molecules dissolved in the seawater. The water molecule found itself surrounded by methane, ammonia, hydrogen, and carbon dioxide, gases that were essential for the prebiotic chemistry taking place in this ancient sea. As it moved through the water, the molecule's polarity allowed it to dissolve various solutes, creating a chemically rich environment. Its slightly negative oxygen atom attracted positive ions, while its slightly positive hydrogen atoms attracted negative ions. This ability to form hydrogen bonds with other molecules made water an excellent solvent, capable of dissolving a wide range of substances. This property was crucial for the biochemical processes that were beginning to take shape. The water molecule's journey soon took it to a region of intense activity, a hydrothermal vent located on the ocean floor. These vents, formed by the movement of tectonic plates, released superheated, mineral-laden water from the Earth's crust into the cold ocean. As the water molecule approached the vent, it was drawn into the turbulent mix of hot and cold waters. The drastic temperature gradients and the presence of catalytic minerals created a unique environment where chemical reactions could occur at an accelerated pace. In the vicinity of the hydrothermal vent, the water molecule encountered a variety of dissolved ions and gases, including hydrogen sulfide, methane, and iron. The high temperatures and pressures near the vent facilitated the synthesis of complex organic molecules from simpler precursors. The water molecule played a crucial role in these reactions, acting as a solvent and participating directly in hydrolysis and condensation reactions. The heat from the vent provided the energy needed to drive these processes, while the minerals acted as catalysts, promoting the formation of organic compounds. One such reaction involved the synthesis of amino acids, the building blocks of proteins. The water molecule was part of the solvent environment that allowed ammonia and hydrogen cyanide to react with other organic molecules, forming amino acids through a series of steps. These amino acids, once formed, could interact with each other, sometimes in the presence of catalytic surfaces, to form short peptide chains. The peptides, in turn, could catalyze further reactions, contributing to the growing complexity of the molecular environment. As the water molecule continued its journey, it was carried away from the hydrothermal vent by ocean currents, moving through cooler waters rich with newly synthesized organic compounds. These compounds included simple sugars, nucleotides, and lipids, each of which played a role in the prebiotic chemistry that was shaping the early biosphere. The water molecule's ability to dissolve and transport these molecules was essential for their distribution and interaction throughout the ocean. In shallow tidal pools, the water molecule experienced cycles of evaporation and condensation. During the heat of the day, water evaporated, leaving behind concentrated solutions of organic molecules. As the temperature dropped at night, the water condensed, bringing these molecules back into solution. This cyclical process promoted the formation of increasingly complex organic structures. The water molecule facilitated these interactions by creating a dynamic environment where molecules could come together, react, and form new compounds. One particularly important class of reactions involved the formation of nucleotides, the building blocks of RNA. In these shallow pools, formaldehyde and glycolaldehyde reacted in the presence of phosphate to form ribose, the sugar component of RNA. The water molecule acted as both a reactant and a medium for these reactions, enabling the formation of the phosphodester bonds that linked the nucleotides together. These reactions were driven by the unique properties of water, including its ability to stabilize charged intermediates and facilitate the removal of water molecules during condensation reactions. The journey of the water molecule also took it into contact with lipid molecules, which spontaneously formed bilayers in the aqueous environment. These lipid bilayers could encapsulate water and dissolved solutes, creating protocells, primitive cellular structures that provided a microenvironment for biochemical reactions. Inside these protocells, the water molecule helped maintain the proper ionic balance and facilitated the interactions between nucleotides, amino acids, and other organic molecules. The formation of protocells was a critical step in the evolution of life, providing a protected space where self-replication and metabolism could occur more efficiently. As the water molecule moved through different environments, it also played a role in maintaining the stability and functionality of macromolecules. The hydrogen bonds formed between water molecules and the polar groups of proteins and nucleic acids helped stabilize their three-dimensional structures. In RNA molecules, hydrogen bonds between complementary bases formed double-stranded regions that were crucial for replication and catalysis. The water molecule's ability to form a network of hydrogen bonds provided the structural integrity needed for these complex biomolecules to function. Throughout its journey, the water molecule participated in countless chemical reactions, each contributing to the gradual increase in molecular complexity. Its role as a solvent, reactant, and stabilizer was indispensable in the formation of the first biomolecules. The unique properties of water, including its high specific heat capacity, high heat of vaporization, and ability to form hydrogen bonds, created an environment conducive to the emergence of life. In the primordial ocean, the water molecule facilitated the formation of a rich chemical landscape where organic molecules could interact and evolve. The combination of energy from sunlight, geothermal activity, and lightning drove the synthesis of increasingly complex compounds. The water molecule's journey through hydrothermal vents, tidal pools, and protocells highlights its central role in the prebiotic chemistry that set the stage for the origin of life. As we delve deeper into the significance of water in the formation and stabilization of early biomolecules, we will explore how its unique properties enabled the assembly of the first self-replicating systems. We will examine the role of water in the formation of hydrogen bonds, the stabilization of macromolecular structures, and the facilitation of key biochemical reactions. Through this exploration, we will gain a deeper understanding of how water, a seemingly simple molecule, was instrumental in shaping the biochemical landscape of early Earth and facilitating the emergence of life. The journey of our water molecule from the ocean to hydrothermal vents and tidal pools illustrates the dynamic and interconnected nature of the early Earth environment. The interactions between water, organic molecules, and minerals created a complex and evolving system where the building blocks of life could form, interact, and replicate. The unique properties of water, including its polarity, solvent capabilities, and hydrogen bonding capacity, were essential in driving the chemical processes that led to the emergence of life. As the water molecule drifted through the ancient seas, it witnessed the gradual transformation of simple chemical compounds into the complex structures that characterize living organisms. Its journey underscores the central role of water in the origin of life, highlighting how this ubiquitous molecule facilitated the synthesis, stabilization, and evolution of the first biomolecules. The story of the water molecule's journey is a testament to the intricate and interconnected processes that shaped the early Earth and set the stage for the emergence of life. Through understanding the role of water, we gain insights into the fundamental principles of biochemistry and the conditions that made life possible on our planet. As we transition to the next chapter, we will further explore the biochemical significance of water, delving into its role in stabilizing the structures of nucleic acids and proteins, its involvement in catalytic processes, and its contribution to the dynamic environment of protocells. This exploration will deepen our appreciation for the central role that water played in the evolution of life and its continued importance in sustaining the diversity of life on Earth today. Through this journey, we will uncover the profound ways in which a simple molecule like water has shaped the complex and wondrous phenomenon of life. Water is often described as the universal solvent due to its unparalleled ability to dissolve a wide variety of substances. This characteristic is critical to its role in the chemistry of life. The unique properties of water as a solvent are central to many biochemical processes, from the formation of the earliest biomolecules to the complex metabolic pathways in modern organisms. Understanding these solvent properties requires an examination of water's molecular structure, its interactions with other molecules, and the biochemical implications of these interactions. The molecular structure of water is simple yet profoundly influential in its solvent capabilities. A water molecule consists of two hydrogen atoms covalently bonded to a single oxygen atom, creating a bent or V-shaped molecule. This shape results from the electron pair geometry around the oxygen atom, which has two lone pairs of electrons. The angle between the hydrogen-oxygen-hydrogen atoms is approximately 104.5 degrees. This bent geometry, combined with the difference in electronegativity between oxygen and hydrogen, gives rise to a polar molecule. The polarity of water molecules is a fundamental aspect of their solvent properties. Oxygen, being more electronegative, attracts the shared electrons more strongly than hydrogen, resulting in a partial negative charge on the oxygen atom and a partial positive charge on the hydrogen atoms. This distribution of charges creates a dipole moment, making water a highly polar molecule. The polarity of water enables it to form hydrogen bonds with other polar molecules and ions, facilitating their dissolution. Hydrogen bonding is a key feature of water's interactions with other molecules. In a hydrogen bond, the partial positive charge on the hydrogen atom of one water molecule is attracted to the partial negative charge on the oxygen atom of a neighboring water molecule. These bonds, although weaker than covalent bonds, are strong enough to create a cohesive network of water molecules. This network of hydrogen bonds contributes to water's high cohesion, surface tension, and its ability to dissolve a wide range of substances. Water's ability to dissolve ionic compounds is particularly important in biological systems. When an ionic compound, such as sodium chloride, is introduced into water, the polar water molecules surround the positive and negative ions, effectively separating them and keeping them in solution. The partially negative oxygen atoms of water molecules are attracted to the positive sodium ions, while the partially positive hydrogen atoms are attracted to the negative chloride ions. This process, known as solvation or hydration, stabilizes the ions in solution and prevents them from recombining. The solvent properties of water also extend to polar molecules. Water can dissolve a variety of polar organic molecules, including sugars, amino acids, and nucleotides. The ability of water to form hydrogen bonds with these molecules is crucial for their solubility. For instance, the hydroxyl groups, or O in sugars, can form hydrogen bonds with water molecules, making sugars highly soluble in water. Similarly, the amino and carboxyl groups in amino acids interact with water, facilitating their dissolution and transport in biological systems. In the context of early Earth, water's solvent properties were essential for the formation and stability of biomolecules. The oceans, rich in dissolved gases and minerals, provided a medium where simple molecules could interact, react, and form more complex structures. The polarity of water allowed it to dissolve and transport a wide range of reactants, creating a dynamic and interactive chemical environment. This environment was conducive to the synthesis of amino acids, nucleotides, and other organic molecules essential for life. The role of water in dissolving and transporting biomolecules extends to its function in modern biological systems. In living organisms, water is the primary solvent in which biochemical reactions occur. The cytoplasm of cells is an aqueous solution that contains a complex mixture of ions, proteins, nucleic acids, and other biomolecules. Water's ability to dissolve and transport these substances is critical for maintaining cellular functions and facilitating metabolic processes. One of the most important roles of water as a solvent is in enzymatic reactions. Enzymes, which are biological catalysts, require a specific aqueous environment to function optimally. The active sites of enzymes, where substrate molecules bind and undergo chemical transformations, often rely on the presence of water for proper orientation and stabilization of reactants. Water molecules can participate directly in these reactions, acting as nucleophiles or proton donors, acceptors, thereby facilitating the catalytic process. For example, in hydrolysis reactions, water molecules break down larger molecules into smaller units. This process is fundamental in the digestion of macromolecules such as proteins, carbohydrates, and nucleic acids. In protein hydrolysis, water molecules attack peptide bonds, cleaving them into individual amino acids. Similarly, in nucleic acid hydrolysis, water molecules cleave phosphodiester bonds, breaking down DNA and RNA into their constituent nucleotides. These reactions highlight the indispensable role of water as a reactant and solvent in biochemical processes. Water's solvent properties also play a critical role in the transport of substances across cell membranes. The selectively permeable nature of biological membranes allows cells to control the movement of ions and molecules in and out of the cell. Water facilitates this process by dissolving the solutes that need to be transported. For instance, ions such as sodium, potassium, calcium, and chloride are transported across cell membranes through ion channels and pumps, all of which operate in an aqueous environment. The transport of glucose and other nutrients into cells also relies on water's solvent properties to maintain solute concentrations and osmotic balance. In addition to its role in dissolving and transporting biomolecules, water's solvent properties are crucial for maintaining the structure and function of macromolecules. Proteins, nucleic acids, and polysaccharides all have specific three-dimensional structures that are stabilized by interactions with water molecules. For proteins, the hydrophilic and hydrophobic interactions with water influence their folding and stability. Hydrophilic amino acid residues tend to be exposed on the protein surface, interacting with the aqueous environment, while hydrophobic residues are typically buried within the protein core. This arrangement is essential for the proper folding and function of proteins. Nucleic acids such as DNA and RNA also rely on water for structural stability. The double helix structure of DNA is stabilized by hydrogen bonds between complementary bases and by hydrophobic interactions between the stacked base pairs. Water molecules surrounding the DNA helix form a hydration shell that helps maintain its structural integrity. Similarly, the secondary and tertiary structures of RNA are stabilized by interactions with water, which are critical for the molecule's catalytic and regulatory functions. Water's role in biochemical reactions is further exemplified by its participation in redox reactions, where it acts as both a solvent and a reactant. In cellular respiration, for instance, water is produced as a byproduct of the oxidation of glucose. The electrons and protons generated during this process are transferred through the electron transport chain, ultimately combining with oxygen to form water. This reaction is essential for the generation of ATP, the primary energy currency of cells. Photosynthesis, the process by which plants, algae, and certain bacteria convert light energy into chemical energy, also relies on water. In the light-dependent reactions of photosynthesis, water molecules are split into oxygen, protons, and electrons through a process called photolysis. The electrons are used to generate ATP and NADPH, which are then utilized in the Calvin cycle to fix carbon dioxide into organic molecules. Water's role as both a solvent and a reactant in these processes underscores its fundamental importance in energy metabolism. The unique thermal properties of water also contribute to its role in biochemical reactions and cellular stability. Water's high specific heat capacity allows it to absorb and release heat without undergoing significant temperature changes. This property is vital for maintaining stable temperatures in living organisms, protecting them from rapid fluctuations that could disrupt biochemical processes. Water's high heat of vaporization enables evaporative cooling, which is essential for regulating body temperature in many organisms. In the early Earth environment, water's solvent properties were instrumental in the formation and stabilization of protocells. Protocells, composed of lipid bilayers, provided a microenvironment where prebiotic reactions could occur more efficiently. The hydrophobic and hydrophilic interactions of lipid molecules with water facilitated the spontaneous formation of bilayers, creating compartments that concentrated reactants and protected them from the external environment. Inside these protocells, water maintained the ionic balance and facilitated interactions between organic molecules, enhancing the efficiency of prebiotic chemistry. The importance of water in the origin of life extends to its role in the synthesis of complex organic molecules. The formation of nucleotides, the building blocks of RNA, involves several steps that require water as a solvent and reactant. For example, the synthesis of ribose, the sugar component of RNA, involves the condensation of formaldehyde in an aqueous environment. Water molecules facilitate these reactions by stabilizing charged intermediates and enabling the removal of water molecules during condensation reactions. The subsequent formation of phosphodiester bonds between nucleotides, which creates RNA strands, also occurs in an aqueous environment. The role of water in the prebiotic synthesis of amino acids and peptides is equally significant. Amino acids can form through reactions between simple molecules such as hydrogen cyanide, ammonia, and aldehydes in aqueous solutions. Once formed, amino acids can interact in water to form peptides through condensation reactions. These peptides can then fold into secondary structures stabilized by hydrogen bonds with water molecules, creating the precursors to proteins. Water's involvement in the formation and stabilization of macromolecules highlights its central role in the chemistry of life. The unique properties of water as a solvent, including its polarity, hydrogen bonding capacity, and ability to dissolve a wide range of substances, created an environment conducive to the emergence of life. Water facilitated the interactions between organic molecules, enabling the formation of complex structures and the evolution of self-replicating systems. As we continue to explore the significance of water in the origin of life, we will delve deeper into its role in stabilizing the structures of nucleic acids and proteins, its involvement in catalytic processes, and its contribution to the dynamic environment of protocells. Through this exploration, we will gain a deeper understanding of how water, a seemingly simple molecule, was instrumental in shaping the biochemical landscape of early Earth and facilitating the emergence of life. The journey of water molecules through various environments, from the oceans to hydrothermal vents and tidal pools, underscores their central role in the intricate web of chemical reactions that led to the origin and evolution of life on our planet. In the vast expanse of the early Earth's oceans, hydrothermal vents stood as towering pillars of geothermal activity, continuously shaping the underwater landscape. These vents, located along mid-ocean ridges and other tectonic plate boundaries, discharged superheated, mineral-rich water into the frigid depths of the ocean. This unique environment created steep chemical gradients and an array of physical conditions that were instrumental in the synthesis of organic compounds and potentially served as the cradle of life on Earth. Hydrothermal vents are formed by the movement of tectonic plates beneath the ocean floor. As these plates diverge, magma from the Earth's mantle rises to fill the gap, creating new oceanic crust. Seawater penetrates through cracks and fissures in this newly formed crust, descending until it reaches the hot molten rock below. This interaction with the magma heats the water to temperatures that can exceed 350 degrees Celsius. The superheated water becomes enriched with dissolved minerals and metals, creating a chemically distinct solution. Eventually, this mineral-laden water is expelled back into the ocean through hydrothermal vents, creating a dynamic and complex environment. The physical and chemical conditions at hydrothermal vents are extreme and varied. The temperature can range from the boiling point of water to several hundred degrees Celsius, depending on the proximity to the venting fluid. The pressure is immense, corresponding to the depths at which the vents are located, often several kilometers below the ocean surface. These high-pressure conditions influence the boiling point of water, allowing it to remain liquid at temperatures that would otherwise cause it to vaporize at sea level. The water expelled from hydrothermal vents is rich in dissolved minerals, including sulfides, iron, magnesium, calcium, and silica. These minerals precipitate upon contact with the cold ocean water, forming towering structures known as chimneys or black smokers. The precipitated minerals create a visually striking contrast between the dark, mineral-laden water and the clear surrounding seawater. These chimneys can grow to significant heights, providing a scaffold for a variety of chemical reactions and microbial life. The steep chemical gradients at hydrothermal vents are critical for driving prebiotic chemistry. The interaction between the hot, reduced vent fluids and the cold, oxygenated seawater creates redox gradients that provide the energy necessary for chemical reactions. This energy can be harnessed to reduce carbon dioxide and other simple molecules, leading to the synthesis of more complex organic compounds. The presence of catalytic minerals, such as iron-sulfur clusters, further facilitates these reactions by lowering the activation energy required for bond formation and rearrangement. One of the key reactions thought to occur at hydrothermal vents is the reduction of carbon dioxide to methane and other hydrocarbons through the Fischer-Tropsch type synthesis. This process involves the catalytic conversion of carbon monoxide and hydrogen into hydrocarbons over metal surfaces, such as those found in iron-sulfur minerals. Laboratory simulations of hydrothermal vent conditions have demonstrated that Fischer-Tropsch type synthesis can produce a variety of organic molecules, including alkanes, alcohols, and fatty acids. These molecules are fundamental building blocks for more complex biomolecules, such as lipids, amino acids, and nucleotides. Hydrothermal vents also provide a continuous source of hydrogen gas, which is a crucial reducing agent in prebiotic chemistry. The serpentinization process, in which water reacts with ultramafic rocks in the Earth's mantle, generates hydrogen gas along with other reduced compounds such as methane and hydrogen sulfide. These reducing conditions create an environment conducive to the synthesis of organic molecules from simple inorganic precursors. The presence of hydrogen gas allows for the reduction of carbon dioxide and other oxidized molecules, facilitating the formation of hydrocarbons and other organic compounds. The unique conditions at hydrothermal vents not only promote the synthesis of organic molecules, but also provide a microenvironment where these molecules can accumulate and interact. The porous structure of the mineral chimneys creates numerous microhabitats with varying chemical conditions. These microenvironments allow for the concentration of organic molecules, increasing the likelihood of their interaction and polymerization. The physical structure of the chimneys also protects the organic molecules from dilution and degradation, providing a stable environment for chemical reactions to proceed. One of the significant advantages of hydrothermal vents as a potential cradle of life is their ability to support the formation of protocells. Protocells are simple vesicles composed of lipid bilayers that can encapsulate organic molecules and provide a microenvironment for chemical reactions. The synthesis of lipids at hydrothermal vents, facilitated by FTT synthesis and other reactions, would provide the necessary components for protocell formation. The mineral surfaces at the vents could also serve as catalytic sites for the polymerization of nucleotides and amino acids, leading to the formation of RNA and peptides within these protocells. The hydrothermal vent environment offers a plausible setting for the emergence of self-replicating RNA molecules, a key component of the RNA world hypothesis. The catalytic minerals present at the vents, such as iron-sulfur clusters and clays, can facilitate the formation of phosphodiester bonds between nucleotides, creating RNA strands. These minerals can also promote the assembly of nucleotides from simpler precursors, such as formaldehyde and hydrogen cyanide. The high temperatures and pressures at the vents provide the energy needed for these reactions, while the mineral surfaces stabilize the intermediate compounds and facilitate their interactions. The potential for hydrothermal vents to serve as a cradle of life is further supported by the presence of modern vent ecosystems, which are rich in microbial life. These ecosystems are based on chemosynthesis, a process by which microbes derive energy from the oxidation of inorganic molecules such as hydrogen sulfide rather than from sunlight. The discovery of these chemosynthetic communities in the late 20th century revolutionized our understanding of life's potential origins, demonstrating that life could thrive in the absence of sunlight, relying instead on the chemical energy provided by hydrothermal vent processes. The microorganisms found at modern hydrothermal vents include a variety of extremophiles, such as hyperthermophilic archaea and bacteria, which thrive at high temperatures and pressures. These organisms possess unique metabolic pathways that allow them to utilize the chemical energy available at the vents. For example, some microbes oxidize hydrogen sulfide to sulfate, while others reduce carbon dioxide to methane or acetate. These metabolic processes are similar to the prebiotic reactions thought to occur at early hydrothermal vents, suggesting a continuity between prebiotic chemistry and early microbial life. The genetic and metabolic diversity of vent microorganisms provides further insights into the potential pathways for the emergence of life. The presence of ancient and conserved genes in these organisms, such as those encoding for ribosomal RNA and key metabolic enzymes, suggests that hydrothermal vent environments may have been among the earliest habitats for life on Earth. The study of these extremophiles offers valuable clues about the conditions and biochemical pathways that could have supported the origin of life. In addition to their role in the synthesis of organic molecules and the formation of protocells, hydrothermal vents also provide a continuous source of nutrients and energy. The vent fluids contain a variety of dissolved ions and gases, such as sulfides, methane, and ammonia, which can serve as building blocks for organic synthesis and as energy sources for metabolic reactions. The dynamic and continuously replenished nature of hydrothermal vents ensures a steady supply of these essential components, creating a stable environment for the development and evolution of early life forms. The potential for hydrothermal vents to serve as the cradle of life extends beyond Earth. The discovery of similar vent-like structures on other planetary bodies, such as the icy moons Europa and Enceladus, raises the possibility that life could exist elsewhere in the solar system. These moons have subsurface oceans in contact with a rocky mantle, creating conditions analogous to Earth's hydrothermal vents. The presence of water, heat, and the necessary chemical ingredients in these environments suggests that the processes leading to the synthesis of organic molecules and the emergence of life could be universal. In summary, hydrothermal vents represent a unique and dynamic environment that played a crucial role in the synthesis of organic compounds and the potential origin of life on Earth. The extreme physical and chemical conditions at these vents, including high temperatures, pressures, and redox gradients, provided the energy and catalysts necessary for prebiotic reactions. The continuous supply of minerals and gases from the Earth's mantle created a chemically rich environment where organic molecules could form, accumulate, and interact. The formation of protocells and the emergence of self-replicating RNA molecules within these microenvironments highlight the vent's potential as a cradle of life. The study of hydrothermal vent ecosystems and the extremophiles that inhabit them offers valuable insights into the conditions and pathways that could have supported the origin of life. The discovery of similar environments on other planetary bodies suggests that hydrothermal vents may be a common setting for the emergence of life in the universe. As we continue to explore the significance of water and its role in the formation and stabilization of early biomolecules, we gain a deeper understanding of the intricate and interconnected processes that shaped the early Earth and set the stage for the evolution of life. Through this exploration, we uncover the profound ways in which a simple molecule like water, in concert with the dynamic environment of hydrothermal vents, facilitated the emergence of life and its subsequent evolution on our planet. Conclusion. In the grand tapestry of the Earth's primordial landscape, water emerges as a pivotal player weaving together the threads of chemistry and biology to create the foundation for life. The journey of a single water molecule, traversing the ancient oceans, interacting with hydrothermal vents, and participating in the formation of early biomolecules, underscores the critical role that water played in the origin of life. This exploration into the multifaceted role of water reveals how its unique properties as a solvent, its involvement in biochemical reactions, and its capacity to stabilize and transport molecules were instrumental in the emergence of the first living systems. Water's molecular structure, characterized by its polarity and ability to form hydrogen bonds, is fundamental to its role as a solvent. The bent shape of the water molecule, with its partial positive and negative charges, allows it to dissolve a wide variety of substances, creating a chemically rich environment where organic molecules can interact and react. This solvent capability is essential for the synthesis and stabilization of biomolecules, from the simplest organic compounds to the complex macromolecules that constitute life. The polarity of water molecules enables them to surround and stabilize ions and polar molecules, facilitating their dissolution and transport. This property is crucial in the early Earth's oceans, where water acted as a medium for prebiotic chemistry. The ability of water to dissolve gases such as carbon dioxide, methane, and ammonia, along with minerals from hydrothermal vents, created a dynamic and interactive chemical environment. In this milieu, simple molecules could form through a variety of chemical reactions driven by energy sources such as geothermal heat, ultraviolet radiation, and lightning. Hydrothermal vents, with their unique combination of high temperatures, pressures, and chemical gradients, provided an ideal setting for the synthesis of organic molecules. The superheated, mineral-laden water expelled from these vents facilitated reactions such as Fischer-Tropsch-type synthesis, leading to the formation of hydrocarbons, alcohols, and fatty acids. These reactions, catalyzed by minerals like iron-sulfur clusters, laid the groundwork for more complex organic compounds, including amino acids, nucleotides, and lipids. The steep chemical gradients at hydrothermal vents, coupled with the presence of catalytic surfaces, promoted the assembly of these building blocks into larger structures. Amino acids could link together to form peptides, nucleotides could polymerize into RNA strands, and lipids could spontaneously assemble into bilayers, creating protocells. These protocells, with their lipid bilayer membranes, provided a protected microenvironment where biochemical reactions could occur more efficiently. The role of water in these processes was indispensable, acting as both a solvent and a participant in chemical reactions. The importance of water extends to its role in stabilizing the structures of macromolecules. Hydrogen bonds formed between water molecules and the polar groups of proteins and nucleic acids help maintain their three-dimensional structures. For RNA molecules, hydrogen bonds between complementary bases formed double-stranded regions necessary for replication and catalysis. The hydration shells formed by water molecules around DNA and RNA provided structural integrity essential for their function as genetic material. Water's participation in biochemical reactions, such as hydrolysis and condensation, was crucial for the formation and breakdown of macromolecules. In hydrolysis reactions, water molecules cleaved larger molecules into smaller units, a process fundamental to the digestion of macromolecules. Condensation reactions, on the other hand, involved the removal of water molecules to form bonds between monomers, facilitating the synthesis of polymers like proteins and nucleic acids. These reactions underscore the dual role of water as both a reactant and a medium for biochemical processes. The thermal properties of water, including its high specific heat capacity and heat of vaporization, provided a stable environment for prebiotic chemistry. Water's ability to absorb and distribute heat efficiently prevented extreme temperature fluctuations, maintaining conditions conducive to chemical reactions. This stability was vital for the accumulation and interaction of organic molecules, supporting the gradual increase in molecular complexity that led to the emergence of life. The journey of water molecules through the ancient oceans, interacting with hydrothermal vents and participating in the formation of early biomolecules, illustrates the dynamic interplay between the Earth's physical environment and the chemical processes that gave rise to life. Water's unique properties as a solvent, its involvement in biochemical reactions, and its capacity to stabilize and transport molecules were essential in shaping the biochemical landscape of early Earth. As we transition to the next chapter, we turn our attention to the formation and importance of early cellular life. The emergence of protocells, composed of lipid bilayers encapsulating organic molecules, represents a critical step in the evolution of life. These early cellular structures provided a microenvironment where self-replication and metabolism could occur more efficiently, paving the way for the development of more complex life forms. The study of protocells and the transition to cellular life offers valuable insights into the processes that drove the evolution of early life on Earth and the intricate mechanisms that underpin biological complexity. In exploring the formation of early cellular life, we will delve into the properties and functions of protocells, the role of lipid bilayers in creating compartments, and the significance of self-replication and metabolic pathways. Through this examination, we will gain a deeper understanding of how the interplay between chemistry and biology led to the emergence of the first living cells, setting the stage for the incredible diversity of life that would follow. The journey from simple molecules to complex cellular systems highlights the profound ways in which the Earth's physical and chemical environment shaped the evolution of life, providing a foundation for the study of life's origins and its continued evolution.